Welcome to our exploration of genetics through taste. Today we'll examine how each of our 8 biology units connect to our PTC tasting experiment, where we tested for bitter taste receptors, collected DNA, and used molecular techniques to identify our genotypes. To begin, Unit 1, Chemistry of Life. Life is built on 4 major macromolecules: proteins, lipids, nucleic acids, and carbohydrates. DNA, our focus today, is a nucleic acid composed of nucleotides, each containing a phosphate group, deoxyribose, and nitrogen base. The bases pair specifically through hydrogen bonding, where A binds with T, forming 2 hydrogen bonds, and G binds with C, forming 3 hydrogen bonds. This complementary base pairing is crucial for DNA's double helix structure and replication. In our lab, we utilize DNA's inherent negative charge from the phosphate groups during gel electrophoresis. When an electric field is applied, DNA's fragments migrate towards the positive electrode through the gel matrix. Smaller fragments move faster and travel farther than the larger ones, creating distinct banding patterns. This size-based separation allows us to analyze different variants for our TAS2R38 gene and determine our specific genotype based on fragment sizes.
All right, so Unit 2 focuses on cell structure and function. To start, all cells are categorized as either eukaryotic or prokaryotic. Eukaryotic cells have membrane-bound organelles, including a nucleus, while prokaryotic cells do not. Key feature of eukaryotic cells is compartmentalization, which allows different cellular processes to occur efficiently in specialized structures or organelles. 2 important components here are the phospholipid bilayer, which forms the cell membrane, and the nucleus, which houses the cell's DNA. On day 1 of our lab, we began by collecting samples. We used the centrifuge to isolate a cell pellet, which contained organelles and the phospholipid bilayer, and just the nucleus within which contained the DNA. We then used a PCR machine to heat and denature the phospholipid bilayer, exposing the DNA so that we could then further analyze it. And the diagram here to the right illustrates the major organelles within a eukaryotic cell.
Next up, Unit 3, Cellular Energetics. Enzymes are biological catalysts that dramatically speed up biochemical reactions by lowering activation energy, which is the barrier that must be overcome for reactions to proceed. They achieve this through the precisely shaped active sites that bind to specific substrates, forming enzyme-substrate complexes. In our experiment, we use restriction enzymes that recognize the DNA sequence GGCC and cleaves both strands between the G and C nucleotides. This enzyme evolved in bacteria as a defense mechanism against foreign DNA. The specificity is crucial because genetic mutations can alter these recognition sites. For example, a single nucleotide change from GGCC to GGGC completely prevents the enzyme from binding and cutting. Different alleles of the TAS2R38 gene contain different numbers of restriction sites. So when we digest our PCR products, we get distinct fragment patterns. Individuals with different genotypes produce different numbers and sizes of DNA fragments, creating the unique banding patterns we observed on our gel.
In Unit 4, we focus on how cells reproduce and how they communicate with each other. First, cells reproduce through processes like mitosis and meiosis, which are essential for growth, repair, and reproduction. Cells also communicate through 3 steps: reception, transduction, and response, during which a signal is detected, the signal is processed, and the cell reacts, respectively. Relating to our lab, we looked at gustatory cells, which are found on the papillae of the tongue. These cells detect taste molecules, and in our lab, seeing as we worked with PTC phenylthiocarbamide, we noted the bitter compound that acted as a ligand or a signal molecule within the lab. When PTC binds to the taste receptors, it triggers signal transduction, which is a chain reaction that sends neurotransmitters to the brain. The result is a response being the sensation of bitterness. And the diagram to the right provides some insight into the idea of taste bulbs and gustatory cells on the tongue.
Now we're going to be talking about Unit 5, which is heredity. Heredity governs how traits pass from parents to offspring through genes, following predictable patterns including dominant, recessive, codominant, and incomplete dominant inheritance. Each individual carries 2 copies of every gene, one from each parent, called alleles. Our genotype represents the specific combination of alleles we inherit, while our phenotype is the observable expression of those genes. The PTC tasting traits demonstrate incomplete dominance, where neither allele is completely dominant over the other. This means individuals with 2 different alleles, called heterozygous, Heterozygotes express an intermediate phenotype between the two homozygous conditions. In practical terms, heterozygous experience moderate bitter taste sensitivity, while homozygous are either strong tasters or non-tasters. Through PCR amplification, we exponentially increase copies of our TAS2R38 gene, making it detectable for analysis. Our gel electrophoresis results directly revealed our genotypes by showing specific banding patterns, demonstrating the molecular basis of inheritance and how genetic variation creates the diversity in human taste perception we observe in populations. Unit 6, Gene Expression Regulation. Gene expression follows a central dogma of molecular biology. DNA is transcribed into messenger RNA, which then is translated into functional proteins at ribosomes. This process transforms genetic information into the proteins that carry out cellular functions. The TAS2R38 gene contains the coding sequence for a G protein coupled receptor protein that spans cell membranes and taste buds. When this receptor encounters bitter compounds like PTC, it undergoes conformational change that triggers intracellular signaling cascades, ultimately leading to nerve impulses interpreted by our brain as bitter taste. During PCR, we use Taq polymerase, a heat-stable DNA polymerase from thermophilic bacteria, to repeatedly replicate our target gene sequence through cycles of denaturization, annealing, and extension. Single nucleotide polymorphism, or SNPs, represent the most common type of genetic variation between individuals. In TAS2R38, these SNPs alter the amino acid sequence of the taste receptor protein, changing its three-dimensional structure and binding infinite affinity for bitter compounds. This demonstrates how even single nucleotide changes can cascade through protein structure and function to create observable phenotypical difference in human sensory perception.
Unit 7 explore the concepts of evolution and how natural selection drives change in populations over time. Natural selection acts as one of the key mechanisms of evolution, wherein traits that improve survival and reproduction become more common in a population over time. As relating to our lab, PTC detection can be seen as favorable. This trait can be favorable because bitterness in nature often indicates toxicity or poison. However, PTC detection isn't necessarily essential for survival in all environments. This is evident since some people are tasters and others are non-tasters, demonstrating that there is genetic variation within our species. We also tested for this by identifying our individual genotypes related to PTC sensitivity. And the diagram here to the right shows microevolution, which refers to small-scale changes within species, in contrast to macroevolution, which demonstrates eventual speciation. PTC sensitivity is microevolution since, based on environments, individual populations spread throughout the world of humans developed PTC sensitivities while others did not. All right, Unit 8 is ecology, which highlights how biotic and abiotic factors shape the survival and behavior of organisms. Every organism interacts with its environment in complex ways and often takes advantage of its specific ecological niche. These interactions influence not only how the organism lives, but how it can evolve over time. Specifically, environmental pressures like temperature, availability of resources, or toxins can affect population genetics. Those individuals best suited to their niche are likely to survive and pass on their alleles. This ties directly to the principles of evolution from Unit 7 and also to the PTC tasting lab, and that some humans can taste the bitter compound while others can't. And this ability to taste may have evolved as a survival mechanism to detect and avoid naturally occurring toxins in plants in certain regions of the world.
De-extinction. The process of bringing back extinct species has moved from science fiction to reality. Colossal Bioscience recently announced the first living dire wolves in 12,500 years using CRISPR gene editing to make 20 precise modifications across 14 genes in gray wolf DNA. This breakthrough raises profound ethical questions. Supporters argue we have more obligation to reverse human-caused extinctions and restore damaged ecosystems while advancing scientific knowledge that benefits all life. However, critics contend resources would be better spent protecting currently endangered species rather than resurrecting the dead. They warn of unpredictable ecological consequences and question whether humans should play God by reversing natural processes. As this technology advances, we must carefully weigh the potential benefits of ecological restoration against the risks of unintended consequences and resource misallocation in our fight to preserve biodiversity.
Okay, so there are bioethical concerns surrounding 23andMe DNA collection. While many people use services like 23andMe to learn about their ancestry or genetic traits, there are deeper issues to consider. For instance, what happens to the DNA data if the company goes bankrupt, which happens with 23andMe? They actually have the ability to sell the DNA information because it's their property now. Pharmaceutical companies, however, already had access to these data records, and unlike medical records, this genetic data is not protected under HIPAA, which raises concerns about misuse and privacy. On one hand, customers do sign user agreements, and the aggregated data has contributed to advances in treatment research, but the situation becomes more relevant when we consider specific genetic traits. Like in PTC genotype analysis, it can show personal data can inform health, but it can also be exploited. So this is something to think about